I want to talk to you about our therapeutic platform of CAR T cells. We're actually focusing on a subpopulation of T cells called an NK T cell, of which I'll talk to you a little bit more about, that have some preferential, some advantages compared to all the T cells that people usually use, but at the same time, they build on the proof of concept that has already been generated with other T cells. We've already seen products on the market with CD19 and a lot more coming behind that. So CAR-NKT is really the platform that we've developed at Medica in collaboration with our partners at Baylor College of Medicine. It is really based on some unique characteristics that NKT cells have compared to some of the other T cells that are being used. We've actually started treating patients already, so our first CAR-NKT product is already in the clinic. I'll talk to you a little bit about that in the slides that are coming up. And we think that the key advantage of NKT cells is really its applicability to off-the-shelf applications. And really, this is where we see tremendous value. We think that this is where the field eventually needs to head towards in order to be able to deliver products of much reduced cost and also being readily available for patients around the world. So I will show you some data very specifically about the lack of GvHD that is associated with these cells, what makes them very important, and really the fact that this all derives from the invariant nature of NKT cells. And by that I mean they all target the same antigens. These are all the same antigens in all of us, so we can exchange NKT cells among us and they will not cause graft versus host disease simply because they only target glycolipid antigens, which are only presented by antigen-presenting cells, and as I said, are the same in every individual. So in some of those, in some ways, it is really a T cell that looks a lot like an NK cell. And so when they were discovered in the beginning, they were actually called NK-like T cells, and that really does show. So what is an NK T cell? This is really where it comes from. An NK T cell really derives from the same progenitor of alpha beta T cells, CD4 and CD8 T cells. It undergoes the same thymic education as your alpha beta T cells. And at some point in this developmental stage, it acquires an invariant TCR that is specific for glycolipids. These glycolipids are most commonly found on pathogens, bacteria, viruses, and some parasites express these glycolipids. And so an NK T cell can only see these glycolipids when they are presented by a specialized type of MHC class I molecule called CD1D, and only antigen-presenting cells express CD1D. So for all intents and purposes, NK T cells cannot see normal tissue. They only see antigen-presenting cells when these are presenting glycolipids. They're similar in some ways to gamma-delta T cells, and as I mentioned, they share some characteristics of NK cells. And for that reason, really, that invariant nature of them, we believe that they are very well suited to off-the-shelf applications. So our platform is basically based on these NK T cells with that invariant TCR. We don't need to remove that TCR, which basically means we get to skip the whole gene editing approach, TALEN, CRISPR, or any other technology. That means the manufacturing process is considerably simpler, and obviously there is a lot less optimization needed in the process development, manufacturing these CAR-NKT products. The CAR that we're using is a fairly standard second-generation CAR, although we have optimized all the subcomponents of the CAR in the NK T cells to make sure that we have the optimal CAR structure on these cells. Together with the CAR, we've added the gene for IL-15 to these cells. We know very well that NK T cells respond very well to IL-15, and so the whole product optimization has been with a CAR optimized in the presence of IL-15, and I will show you some data that really shows that that combination really is significantly better than just using a CAR. And in addition, since we don't actually have to worry about the problem number one of off-the-shelf therapeutics, cellular therapeutics, which is the GVHD, then we decided to focus on problem number two, which is rejection, and that will happen with all off-the-shelf or allogenic cells that are being delivered. And in order to avoid the rejection, or at least delay the rejection as much as we can, we are knocking down MHC class 1 and MHC class 2. And we're doing that by using shRNAs that are incorporated in the same vector as the CAR and the IL-15. And the reason really that we're knocking them down and not out completely is that if we knock out MHC class 1 completely, then the cells will be rejected by NK cells in the patient. So we'll end up with a population of cells that will be rejected by T cells because they have too much MHC class 1, and they will see that some of them will be rejected by NK cells because they have no MHC class 1, but there will be some cells in the middle that will not be rejected, or at least will not be rejected immediately, and will persist for longer. And really what we're trying to achieve there is get enough persistence, because we do know, at least based on the clinical trials that have happened already, that persistence does correlate with efficacy. So this is really how we are attempting to solve that problem. This is a demonstration in an animal model of neuroblastoma. This is a CAR that targets GD2 and also carries IL-15. So the cells are secreting IL-15 locally. And all I'm showing you here is in this animal model, you're seeing here the untreated and untransduced populations. With the CAR alone, you're getting obviously a significant improvement in survival of these animals. But then the addition of IL-15 really makes a huge difference in terms of survival. And so really based on this early data that was generated at Baylor a few years ago, we made the decision that really the platform does have to have IL-15 in it in order to be able to achieve the maximum possible efficacy. This is a slide also generated at Baylor that shows the lack of graft versus host disease by injecting these cells into mice. It's actually the same animal model, the GD2 CAR against neuroblastoma cell lines implanted into these mice. And on the top, what you're seeing is CAR T cells that have been transduced with the same CAR as the NK T cells. These come from the same donor, the T cells and the NK T cells. And what you're seeing on the top is a liver that's really significantly necrotic. It's very white. It's not perfused sufficiently. There is a very heavy infiltration of T cells in the micrografts in both the liver and the lung. And compare that with the NK T cells, where you see a very healthy red liver and absolutely no infiltration of T cells in these tissues. I must point out that these mice were sacrificed at about 4 or 5 weeks simply because the CAR T treated mice were experiencing severe GVHD. We've actually kept, in this experiment, we've kept cells in mice for about 11 weeks. We did not see GVHD. Subsequently, we have mice that we have treated for 6 months. And there's still no GVHD while these mice actually control the tumor, whereas untransduced or NK T cells without the IL-15, we do see the tumors coming back. So really this is an effect of the efficacy of the cells and the lack of the GVHD in the mice for significant periods of time. And this is our NKT platform, and I'm really only focusing on the NKT component of our platform now. We have some additional TCR products that we're working on, but I wanted to focus on the NKT. I did mention that we have a GD2 product already in the clinic. We recruited our first patients around the end of August, so it's very recent development for us. The first product is autologous, and that's for the very simple reason that it was easier and faster to go through the regulatory hoops and start the clinical study. We felt that what we needed most is some proof of concept of the platform. We chose GD2 and neuroblastoma because we know neuroblastoma does express very high levels of GD2. It's a validated target. There is an antibody therapeutic on the market against GD2 for neuroblastoma. Unfortunately, this antibody is associated with significant levels of peripheral neuropathy, the significant pain, which is quite unacceptable for these patients, which are in their majority pediatric patients. And in fact, the antibody has to be administered in combination with morphine simply to alleviate that pain. We haven't seen any toxicities with a CAR targeting GD2 in the past at Baylor. We're pretty confident that the toxicity is associated with the antibody rather than the targeting of the target. So this is already in the clinic. We're quite excited about generating some clinical data closer to the middle or second half of next year. It's a dose escalation study looking at 4 different doses, and we will hope to be able to generate a lot more understanding on this platform for solid tumor applications. The second one, and probably a more exciting one, at least in terms of it being a significant milestone for the company, is an allogeneic product against CD19. And while I do recognize that CD19 is probably not the most innovative target out there, we're actually using that because we know it works. And we don't want to have to worry about the target at the same time as generating data on an allogeneic product. We understand a lot more about this target. We know a lot more about the persistence of the autologous products in the patients based on the clinical trials that other companies have performed in the past. So we can really benchmark the findings of our study against what everyone else has seen. This is really, we're estimating that we should be able to start treating patients at the first half of next year. We're probably looking as a conservative timeline, having an IND submitted somewhere around the beginning of next year, and looking at being able to start treating patients within 3 months of that. And that is going to be our first allogeneic product in the clinic, and really generating some proof of concept for the platform. We are working on 2 additional targets, both of which also come through our collaboration with Baylor. GPC3 is a well-validated, understood hepatocellular carcinoma target, and CSPG4 is one that's being developed by Gianpietro Dotti during his time at Baylor and continuing to work on that now at the UNC against glioblastoma and triple-negative breast cancer and a few other solid tumor indications. And again, those we're generating preclinical data expected around the second half of next year. So I'll just summarize, and this is really the slide that tells you everything you need to know about us. We're very excited about the CAR-NKT platform and its potential for off-the-shelf. We are targeting solid tumors as well as hematological malignancies, although in the future we are going to be focusing as much as we can on solid tumors, as you've seen. We're working with world-class universities in developing these platforms. The CAR-NKT platform comes from the Baylor College of Medicine. We're working very closely with UCL and with the cell therapy catapult very, very closely on a TCR platform. Our manufacturing is partly at Baylor, but we've also recently onboarded on the facility of the cell therapy catapult in Stevenage, north of London. We're very happy to be there. It's actually a fantastic setup for us. And just a final word on our investors, Woodford IP Group, which has recently merged with Touchstone Innovations to become one of the largest life science investors in the UK, and Invesco Perpetual, all of them non-traditional VCs, but evergreen funds, which have continued to finance the company forward. And I'll leave it there. Thank you very much.